In this particular session, we're going to look at photosynthesis. In particular, we're going to look at plant photosynthesis. We're not going to go into extremely great detail, but I want you to get a feel for what photosynthesis is doing and what's involved in it. In particular, I'd like you to understand how a plant is designed for photosynthesis from its roots all the way up to its leaves, the entire plant. And to that end, because I think that's important, I've actually designed a lab for you to do on this topic of photosynthesis. What I'd recommend is that you watch this particular video and do the lab, the part that involves looking through the microscope and that sort of thing, then re-watch this video as you are or before you actually answer the questions in the lab. I think when you put all that stuff together, you'll begin to get, and I don't want you to miss, this incredible design of the entire plant to actually produce food for the rest of the animals and things of the land. So in the process of photosynthesis can be summarized by a chemical equation. Sunlight plus carbon dioxide plus water will yield what is a chemical formula for sugar, glucose, plus oxygen. Now that might be something you don't understand. You don't understand this equation, but just maybe think of it as an algebra equation. Maybe you've got that. You've got x plus y equals 3. You've got 3x plus 4y equals 6. You can actually add those equations together and get a third equation. The third equation only looks like one equation, but actually hidden in that third equation are the two equations you added to get there. This is an example of the same sort of thing. This might look a little complicated to you, but actually it's really, really simple because it's hiding a number of hundreds of other equations that have been added together to get this particular equation. And so we're going to look a little bit at the complexity that's hidden in this equation. For now, we want to look back, kind of step back at a 35,000 feet view of this and consider just broadly what this means. What this means is that plants take sunlight, carbon dioxide from the atmosphere, water from the ground, and somehow combine those things to get sugar. And then they toss off some oxygen as a waste product. So for photosynthesis to occur most efficiently, we might think, let's see, I would love to be in a place where all three of the things that I'm using, sunlight, carbon dioxide, and water, are all together in the same place. And that's actually possible in the water. Let's say you're floating around in a lake. You've got the sunlight coming into the top of the lake. You've got carbon dioxide right there in the atmosphere and in the water. And you've got water that you're in. That's a no-brainer. Algae would do photosynthesis right there on the top of a lake. But let's say you're on the land, and we're talking about plants at this moment, so we're asking where would plants do their photosynthesis? You've got the sunlight up above, you've got the carbon dioxide in the atmosphere, but the water's in the ground. The problem on the land is access to water. So there's no place that's really where all three of these things are put together in the same place. So probably the best thing to do is to put it in a place where at least we have two of the three things, and then we're going to have to transport the third thing to that location. And so with plants, what actually happens is that plants actually do their photosynthesis in their leaves, where they're up in the sky, if you wish, in the presence of the sunlight. They're in the midst of the carbon dioxide of the atmosphere. Then they only have to pump the water up from the ground to get it to the location of photosynthesis. So the very first observation is that photosynthesis occurs in the upper part of the plant that's exposed to the sun, in fact, the leaves of the plant. Then we have to, with that understanding, a challenge is going to be, and this is the challenge for the design of plants, to get that water up to the leaves where photosynthesis occurs. So we'll start with that challenge. How is the plant designed in such a way as to maximize that motion of water up from the roots? You see that if you pull up a plant from the ground, you've got a bunch of roots. You've got a number of individual axes that break into smaller and smaller and smaller axes. You've got actually very fine roots if you pull it up carefully, or if you dig around it and then dissolve away the dirt so you can see this. Very, very fine roots. We've got a lot of surface area in those roots to bring in a lot of water. You've had a surface area volume lab where we've talked about the importance of surface area. Surface area is the outside of these individual rootlets. In order to supply the needs for photosynthesis in the leaves, we've got to bring in a lot of water, so we need a lot of surface area. So whereas the roots do have a function to hold the plant in place, the primary function of the roots and the fine structure of the roots is to increase surface area to bring in more water. They wouldn't need all these tiny little fine roots if they really were just using roots to hold themselves in place. We can see that the fineness of the roots is to bring in water. But that isn't enough. Actually, if you look at the roots under a microscope, which you're going to do in the lab, you'll find that when you blow these tiny little roots up, these root hair, these things that look like hair-sized strands, the fine roots up, you find that towards the end of the root, there are these what we call root hairs. Microscopic extensions out of the root that increase the surface area so much more to allow for the absorption of even more water. And I've even referred to, this is not something we're going to see in the lab, but I've even referred in previous lectures to the fact that we also have fungi, that mycorrhizal fungi, that actually invade the root and have similar things as these root hairs reaching out into the soil, gathering even more water for the plant. Collectively, this is how we get the water into the plant. Maximize the surface area of the roots, root hairs, and even mycorrhizal fungi. Now, once we have the water in the plant, it's still sitting down here in the roots. We have to get it now all the way up to the leaves. And here is where we have an extraordinary design of the stem of the plant and the roots themselves, the stem of the plant, and even the leaves themselves to transport water up into the leaves. We have, and the primary actor in this incredible drama is a cell known as a tracheid cell. You're going to look at this under the microscope in the lab. You're going to look at these things to get an impression of what's going on here. A tracheid cell is a long tube-shaped cell that grows and produces a nice cell wall along its length and then evacuates. It actually kicks out the cell contents, leaving just the tube. And then other cells around the tracheids will maintain that cell wall, and the tube is open so that water can go through it. And so it's basically a pipe for water. It's interesting that if you think of, I want to water my garden, so I want to get water from the water source to my garden, we found the most efficient way to do that is to put the water through a tube. A circular tube is the shape that in fact gets the most water through the tube using the least amount of tube material to make it happen. That's pretty cool. So that the shape of tracheids, you'll see, tends to be circular in cross-section and extremely long. That's a design element. It's made to maximally, to in fact, transport the maximum amount of water with the least amount of cell wall material. But there's another feature here that's interesting. I've referred in a previous lecture to a characteristic of water called capillary action. The fact that if you have let's say straws of different sizes, that water goes different distances up into the straw, automatically the water climbs the walls of the straw farther up in smaller tubes. The narrower the straw, the smaller the diameter of the tube, the farther up the water goes. These tracheids are really small tubes or straws. You're going to see that in the lab. You're going to measure the size of these, how small they are. And I mean, it's really, really small. That means that water automatically travels a good distance up these particular tubes without the plant having to do any work in pumping the water up to those locations. They can get up several feet in this way. So until you get to the size of something like a tree, you can actually transport all the water that a plant needs through these tracheids. That is just one cool design. Now, the individual cells, tracheids, are connected end to end, and you got to pass the water from one tracheid cell to the next. Sort of like you got segments of hose that you screw together, and you can get the hose to go a long distance. These individual cells are not quite long enough to go all the way up to the leaves, so we connect them end to end. Where they're connected, they have a plate on each cell so that water goes through the plate, and there's holes in the plate. And interestingly enough, we don't just put if you got a tube here, we don't put the plate in perpendicular to the axis of the tube. We put the plate in at an angle and put holes in it. That actually allows the maximum amount of water to move from one tube to another. Once again, an incredible design. Humans, if they designed it, would have eventually landed upon this kind of design to get the maximum amount of water from the roots to the leaves. So that there is a continuous then chain of tracheids from the roots and from the rootlets through the stem into the veins of the leaf, all the way into the place where photosynthesis is to occur. That's how we get the water into the leaves themselves. Now, once the water gets into the leaves, and it's coming in through the main veins of the leaf, we don't want to lose it. There's been an incredible design to get the water up to the leaves at this point, so we want to make sure that once the water gets into the leaves for the photosynthesis, we're not wasting it away. And so the leaf is designed in an extraordinary way to keep the water in. So the surface of the leaf is covered with special cells that are close packed together in such a way that they tend not to let water out. In between the close packed, what we call epidermal cells of the leaf above and below, in between here, the cells are loosely arranged with space for water to flow around in between. But these epidermal cells are very closely knit together to prevent water from getting out. And in addition to that, there's a waxy coat that is actually superimposed on the leaf, on the epidermal cells, on the top side and the bottom side. Remember, wax is a molecule that repels water, doesn't want water to go through it. So if you put a waxy coat above and below, water that happens to get in between the epidermal cells won't go through that barrier to the outside. This keeps the water in the leaf. Really cool design once again. But we have a problem if we do this really well. If we seal the leaf so that water can't get out, then we also seal the leaf in such a way that carbon dioxide can't get in. And so in order to solve that particular problem, the leaf is actually designed with little doors or holes called stomata in the epidermis, usually on the underside of the leaf, that can open and close. So when the leaf needs the carbon dioxide, because photosynthesis is occurring, so that might happen during the day, for example, it opens up its holes, its stomata, and there's usually, the stomata is formed by two cells on either side of the hole. The cells can pull in water and swell up and close the hole, or they can evacuate water and shrink down and open the hole. Really cool design on opening and closing the stomata. The stomata then allows carbon dioxide in, allow carbon dioxide in, and photosynthesis can occur. Then at night, when photosynthesis, for example, isn't occurring, or when other things make photosynthesis impractical, we close the stomata so that we don't lose the water out of the leaf. Again, you're going to see these things. You're going to see stomata in the lab under the microscope, and it really looks cool. The epidermal cells look cool, so I hope you enjoy all of those things. Now, once we get into the leaf itself, inside the leaf, we now have special cells that do the photosynthesis. They're large cells with a bunch of these chloroplasts. I've introduced the chloroplasts to you already. It's this kidney bean-shaped organelle that has three membrane layers, two on the outside and one really complex one in the middle. It sort of looks like stacked coins or stacked pancakes. Lots of surface area on that internal membrane called the thylakoid membrane. It's got lots and lots and lots of surface area so that it can be the foundation or the floor for all of these molecules that are doing photosynthesis. These machines, the machinery that's involved in photosynthesis is set into the floor of this thylakoid membrane. It has to be a huge membrane, lots of surface area, because photosynthesis involves lots of molecules doing lots of things. You're going to look in the lab, you're going to be looking at chloroplasts in cells, getting a feel for their size relative to the cells. Cells can have lots of chloroplasts in them. Some just have a few, but some can have hundreds of chloroplasts in the cell. And remember, photosynthesis doesn't occur in the cell as a whole. Photosynthesis occurs in these tiny little chloroplasts inside those cells. Now, this is an overview kind of picture of the steps of photosynthesis. Photosynthesis is a very complex process using lots of steps. And an interesting personal experience when I was in graduate school. Soon after I got into graduate school, I'm living in a graduate dorm. In other words, it's a dormitory with lots of other graduate students there. I thought, you know, it would be really cool to go around and knock on the door of the other graduate students and find out what they're working on. Because I bet they're working on all sorts of really interesting, different things that are different from what I'm working on. And when I did that, the first door I knocked on, right across the hallway, was an individual who introduced himself, and when I asked him what he was doing, he says, "I'm working on photosynthesis." And naive me, I said, "Don't we already understand that?" It's like I didn't realize how ridiculous that question was. He said, no, we don't. We don't even know how many steps there are in photosynthesis. He said, we estimate that there's something in excess of 500 chemical steps, and we understand about 50 of them. That's 10%. He says, so my thesis is concentrating on one of those steps, one of those 500 steps we don't understand, and I'm hoping that I can solve that or understand that step. But he says there's also about 60 researchers around the world that have devoted their entire lives to studying photosynthesis, and they're each working on different steps in photosynthesis. They might spend their entire life figuring out one of those steps. Wow, I was impressed. The fact that this process is so complex, we don't even know how many steps there are. There's a lot of them represented in this diagram. In general, photosynthesis is broken up into two series of steps. There's one series of steps that is called the light, is referred to as having the light-dependent reactions. This portion of photosynthesis, with lots and lots of steps, requires light. It is light dependent. There's a second set of steps in photosynthesis that does not require light. It's a light-independent set of reactions. In some plants, interestingly enough, these two things are done in different places. In fact, they're done in different cells at different times. In other cells, they're done in the same cells, the same chloroplasts, at about the same time simultaneously. So photosynthesis can play this game in several different ways. We're going to first look at the light-dependent reactions with a little more detail. Here's a diagram that tries to show in a cartoon fashion some of the major steps in the light-dependent reactions. Not all the steps. This is a summary, again, trying to get some of the simple ones. Notice, first of all, that we're sitting in a phospholipid bilayer membrane that I've referred to before. This is that thylakoid membrane, the thing that looks like stacked coins or stacked pancakes. And these various things stuck in the membrane are proteins that are the machinery of photosynthesis. And again, they're arranged in a particular order because photosynthesis is done step by step, and the membrane makes sure that everything's lined up in the proper fashion. And one of the major, or two of the major components of photosynthesis are these things called photosystems. Photosystem 1, photosystem 2, and to look just briefly at what's going on in a photosystem, we need to step back and consider the nature of light coming from the sun. If you take light from the sun and put it in a prism, you recognize that if it'll splay out on the wall or a piece of paper as a spectrum of light. Red, orange, yellow, blue, green, indigo, violet. You've got a spectrum that light that is impinging on a surface, that's coming in on a surface, is coming in white, meaning it's got all the colors in it. There's a bunch more that we can't see on either side, but the ones we're going to concentrate on are the ones that we can actually see. So if it's a white piece of paper, or if it's a black piece of paper, the light coming to the paper is always white. It's got all the colors. And then what reflects off the paper is what you actually see. You don't see the light that's coming in, you only see the light that comes off the paper. So if you got a white piece of paper, what you're seeing is white light reflected off the paper, which means all the colors are reflected off the paper. If you got a black piece of paper, it means that the piece of paper has absorbed all of the colors. You're getting no light reflected off of the paper. Okay, so you got that so far. Now here's where it gets kind of weird. Let's say you have a red piece of paper. What does that mean? The same color light is coming in, that's all the colors, but what's reflecting out is red. What does that mean? That means that the paper is absorbing all the other colors and reflecting red. So that means that a red piece of paper is not red. It's rejecting red. It's like the opposite of red. It absorbs everything else and kicks the red out. That's kind of, the first time I tried to understand that, that messed up my brain a little bit. When you see a green leaf from a tree, means that light came into that leaf, but it reflected green. It kicked green out. It rejected green. It absorbed the other colors. So photosynthesis in a leaf is somehow picking up or absorbing the colors other than green. That's the energy that it's using to in fact do photosynthesis. And in fact, when you have that spectrum of colors and you consider let's say you take the leaf and you break it up, and this is something you can do in a lab. We won't be doing it in our lab, but it's something I did in organic chemistry lab. We took some leaves and we basically scrambled them up, and then we separated the different molecules in the leaf, and we found a number of what are called pigments in the leaf. A pigment in general, let's say you've got some paint, you want to paint the walls, and you get some base paint, so basically a white paint, and you put a pigment into it. The pigment gives color to the paint. Let's say you want a red wall. You put a red pigment in there. So what is a pigment? A pigment is some molecule, some substance that absorbs certain colors and reflects the color that is the pigment, whatever the pigment is. You got a red pigment, it's reflecting red, it's rejecting red, it's absorbing the rest. If you grind up a leaf and look at its contents, you'll find a number of pigments. Not just one, there's actually a whole bunch of pigments in there. Examples are here. There's a pigment called chlorophyll A, there's a pigment called chlorophyll B, and in fact, plants use photosynthesis, AB photosynthesis, which refers to the fact that chlorophyll A and chlorophyll B are important constituents among the pigments of the leaf. There's also some other pigments called carotenes. Carotenes are derived from carrots are orange, so they tend to be orange pigments. If you take in one of the individual pigments, like chlorophyll A, and look at what happens when light shines on it, what colors reflect, what colors are absorbed. Here you have a diagram that indicates, for chlorophyll A, this white line indicates what colors it absorbs. Chlorophyll A absorbs red, lots of red, doesn't absorb anything really in the yellows and the greens and the blues, but towards the blue-violet, it absorbs most of those colors. Chlorophyll B, it's a little displaced from chlorophyll A. It doesn't do so much absorption of red, but absorbs the orange, and then a lot of the blues. So between the two of them, they really take out the oranges and the reds, and the blues and the violets. Carotenes, again an orange color substance, they take out the blues and even a little bit into the green. When you put all these chlorophyll, all these pigments together, there's this middle range, in particular the green, that isn't absorbed by them, thus reflected by the leaf. So these pigments, when they're all considered together, absorb most everything except the green, and thus you have green plants, green leaves. These photosystems have a bunch of pigments in them that are intended to absorb certain wavelengths of light. The other concept I need to make sure you understand is that when you absorb light, you're absorbing energy. Let's say you go out into the sunlight with a black shirt on. It's warm very quickly. Walk out with a white shirt, it's not so hot. Why? Well, it actually makes sense because remember, the white reflects the light, the black absorbs the light, so black is actually absorbing, your black shirt is absorbing the light from the sun, that's why it's hot. Whereas the white reflects it, so it's not as hot. So with that thought in mind, you recognize that when you do absorb light, you're absorbing energy from the sun. And that's really what the photosystems are doing. They're absorbing light energy from the sun. That energy is what we're eventually, through photosynthesis, going to stack into or pack into a molecule to store energy. Remember, the result of this is sugar that I've already referred to as a molecule that stores energy. So what photosynthesis basically does in its simplest form is it takes energy from the sun, sticks it into a sugar molecule, storing it as energy. The first step in that process is to absorb energy from a photosystem. Energy that is absorbed from photosystem 2 is used to get a little electron really excited. He's bouncing around kind of like a hot potato. He tosses the electron through a series of proteins known as an electron transport chain. You're transporting the electron along a chain of molecules, kind of like a bunch of people sitting in a row, and you throw a hot potato at the one at the end of the row. He throws the potato to the next one, and they just keep on going. Now, each time the potato loses a little bit of energy, cools down along the process. This is designed in really, really cool way so that each time the electron is passed from one molecule to another, it loses just a little bit of energy in a very special way. Just enough energy to allow the electron transport chain to do something really cool, which is to transport hydrogen ions from one side of the membrane to the other. I've already referred to the phospholipid membrane as something that lets water through and oxygen through, but does not let ions through. Hydrogen ion, if it's on one side of the membrane, is not going to spontaneously go through the membrane. But what this electron transport chain does is it takes the energy from the electron, from that excited electron, and uses it to pump or to throw the hydrogen ion from the outside of the membrane to the inside. The effect of this over time, by the way, I'm really simplifying this stuff, oversimplifying this stuff. This is basically what's happening. We build up a bunch of hydrogen ions on the inside of the membrane. What we're doing is kind of analogous to putting water behind a dam. There's a really cool hydroelectric dam near Chattanooga that just fascinates me. It's on what's called Raccoon Mountain. What they do, what the Tennessee Valley Authority does here, is at night, when electricity isn't used as much by people, they buy their own electricity at the lower prices because it isn't as used at that time, and they use it to pump water up from the Tennessee River all the way up to the top of the mountain and store it in a lake at the top of the mountain. Then during the day, when electricity prices are higher, when the rates are higher, they run the water down through a hydroelectric plant producing electricity. So crazily enough, they buy the electricity to pump the water up, sell the electricity as they let it fall down, and they make money at this process. That's just really cool. But the idea is you put water up at a place where, because it's sitting so high, it's got a bunch of energy. It wants to go down, and you use that tendency, because of the second law of thermodynamics, to go down and to turn turbines to produce electricity. This is doing basically the same thing. You're pumping hydrogen ions into this inner compartment here, kind of like pumping water up to the top of a mountain. Now the hydrogen ions, there's a whole bunch of them inside. They want to get out because there's a low concentration out here. Second law of thermodynamics says, from a lab you had earlier, that the hydrogen ions really want to get from one side of the membrane to the other, but they can't because, of course, they're ions, so they can't go through the membrane. Eventually, they get such a concentration high enough that they find their way through this little fella right here. This is an ATP synthase protein. It's got a little channel running through it that's just big enough for the hydrogen ions to get through. And they see their way, ooh, I can get out of here. Second law of thermodynamics says, man, I'm going to go. And so they go right on through that. Now, as they do, there's a little turbine in there, very similar to the turbine that is moved by water coming through a hydroelectric plant. And the turbine actually, when it's turned by these hydrogen ions coming through, it actually takes something called adenosine diphosphate, ADP, and a phosphate, sticks them together, puts energy into that bond, producing what we call ATP, adenosine triphosphate. ATP is like electricity in the cell. It's the source of energy for the cell. This cool process produces a bunch of energy in almost exactly the same way as water is used in hydroelectric plants to get electricity. The result of all of this, again, there's a lot more complexity than what I'm talking about here, but basically what we get at the end of this is a bunch of ATP, and ultimately that little electron the energy was used for this whole process. we end up with some electrons at the end of this and some energy. That's going to be taken over to the light-independent reactions to do the next step. But that's for a future video. That's for the next video. For now, what we've done is we've looked at the light-independent or light-dependent reactions of photosynthesis. It's an amazing design. We'll now go to the light-independent reactions of photosynthesis.